Welcome to this lecture on the fundamentals of biomolecules. Today, we are going to explore the fascinating world of sugar molecules, specifically focusing on anomers, pyranose, and furanose. Understanding how these simple sugars fold and lock into specific shapes is the absolute foundation of carbohydrate biochemistry, and it explains everything from how we store energy to why wood is so strong. To begin our journey, imagine a long, flexible snake that suddenly decides to bite its own tail to form a circle. In the world of biochemistry, sugar molecules like glucose and fructose do exactly the same thing when they are dissolved in water. Instead of staying as straight, rigid chains, these molecules bend around and react with themselves to form stable rings. This ring-forming behavior is absolutely crucial because the three-dimensional shape of a molecule determines its ultimate function in our bodies. In aqueous solutions, like the blood flowing through your veins, these ring forms are thermodynamically much more stable than the open chain forms. Now that we know sugars form rings, let us look at the specific shapes they take. Depending on how the sugar folds, it can form either a six-sided ring or a five-sided ring. A pyranose is a six-membered ring structure consisting of 5 carbon atoms and 1 oxygen atom, named after the chemical compound pyrin. On the other hand, a furanose is a 5-membered ring containing 4 carbon atoms and 1 oxygen atom, named after furan. These are the true functional forms of sugars in nature. For example, the furanose form of ribose is the structural backbone of DNA and RNA, meaning our very genetic code relies on the stability of this 5-membered ring. Understanding the shapes leads us to ask how these rings actually form chemically. These rings form through an intramolecular reaction, meaning the molecule reacts with itself. Specifically, a hydroxyl group on the sugar chain attacks the carbonyl group of the exact same molecule. This reaction creates a new functional group, which we call a hemiacetal for aldose sugars, or a hemiketol for ketose sugars. Importantly, the carbon atom that was originally part of the carbonyl group becomes a new chiral center. This means it is now attached to 4 different chemical groups, which will become very important when we discuss anomers later on. Since this ring formation is a chemical reaction, we can measure its stability using an equilibrium constant, KEQ, which is the ratio of closed rings to open chains. Building on that, we often want to know exactly what percentage of the sugar is in the ring form. This formula converts that equilibrium ratio into a straightforward percentage. By taking KEQ, dividing it by KEQ plus 1, and multiplying by 100, we find the exact proportion of closed rings. When KEQ is a large number, this fraction gets very close to 1, meaning almost all molecules have folded into stable rings. Before we move on, let us see this with a concrete example using D-glucose. We are given that the equilibrium constant for the formation of the glucopyranose ring is 400. We want to find the percentage of glucose molecules that exist in the closed ring form. We substitute 400 into our formula, giving us a fraction of 400 divided by 401. Multiplying by 100 gives us approximately 99.75%. This physical result means that out of every 10,000 glucose molecules in your blood, 9,975 are in the pyranose ring form, and only 25 are open chains. Now that we have proven, mathematically, that sugars exist almost entirely as rings, let us look closer at how they close. Let us go back to our analogy of the snake biting its tail. When the head connects to the tail, it can twist in one of two ways. It can lock into place, facing downwards, or it can lock facing upwards. In sugar chemistry, when the straight chain closes into a Pyranose or Furanose ring, the newly formed connection can also lock into two distinct three-dimensional orientations. We call these two versions anomers. They are slightly different versions of the exact same sugar, but as we will see, this tiny twist changes everything. To define these twists scientifically, we must look at the anomeric carbon. Anomers are a specific type of stereoisomer known as diastereomers. They differ in configuration only at the hemiacetal or hemiketal carbon, which we call the anomeric carbon. When the ring closes, the hydroxyl group on this carbon can point in one of two directions. If the hydroxyl group points in the opposite direction to the reference group, we call it the alpha anomer. If the hydroxyl group points in the same direction as the reference group, we call it the beta anomer. This locks the molecule into its final three-dimensional shape. You might wonder why such a tiny structural difference matters. The distinction between alpha and beta anomers is actually one of the most profound concepts in all of biochemistry. When glucose molecules link together using alpha anomeric bonds, they form starch and glycogen. These are flexible energy storage molecules that our bodies can easily break down and use for fuel. However, when glucose molecules link together using beta anomeric bonds, they form cellulose, the rigid structural material found in plant cell walls and wood. Humans do not possess the enzymes required to break beta linkages, which is exactly why you can eat and digest a soft potato, but you cannot digest a wooden plank. Because these two anomers have different three-dimensional shapes, we can measure them in a lab because they bend polarized light differently. This formula calculates the specific rotation of polarized light for a solution containing a mixture of both alpha and beta anomers. The term X-alpha represents the mole fraction of the alpha anomer, while the alpha symbol in brackets is its pure specific optical rotation. The same applies to the beta terms. If the fraction of the beta anomer increases in the solution, the overall specific rotation of the mixture will shift closer to the pure value of the beta anomer. Let us apply this formula to a real laboratory scenario called mutarotation. We're given that pure alpha-D glucose has a specific rotation of positive 112.2 degrees. Over time, it converts into beta-D glucose, which has a rotation of positive 18.7 degrees, until an equilibrium mixture is reached at positive 52.7 degrees. We set up our equation, replacing the beta fraction with 1 minus the alpha fraction. By distributing the numbers and solving the algebra, we find the alpha fraction is 0.364. This means the mixture is 36.4% alpha and 63.6% beta. Physically, this proves the beta anomer is more stable because its bulky groups are spread further apart. Now that we have calculated their stability, let us look at the actual structures of these two molecules. On the left, you see alpha-D-glucopyranose, and on the right, beta-D-glucopyranose. Focus your attention on carbon number 1, the anomeric carbon on the far right of each ring. In the alpha anomer, the hydroxyl group points downwards, which is opposite to the heavy reference group pointing up on carbon 5. In the beta anomer on the right, that same hydroxyl group points upwards in the exact same direction as the reference group. This visualizes the steric clash we just discussed. The beta form keeps those bulky groups out of each other's way, making it the more stable configuration in nature.
To wrap up our lecture, let us review the most important ideas we have covered. First, sugars in aqueous environments do not remain straight. They spontaneously fold into highly stable Furanose or Pyranose rings. Second, the very act of closing this ring creates a new chiral center at the anomeric carbon, locking the molecule into either an alpha or beta anomer. Finally, these seemingly tiny structural differences dictate the physical properties of the sugar, determining whether it will be used for flexible energy storage, like starch, or rigid structural support, like cellulose. You now have the fundamental key to unlocking the complex world of carbohydrate biochemistry.